So it is 9 p.m. and I have just finished my last time point for this enormous time course day, and I am finally headed home.
Yep, that's me. You're probably wondering how I ended up in this situation.
Hi everyone and welcome back to another video. My name is Jane and I'm a second year PhD candidate in cancer research. So today is Sunday.
Hello darkness, my old friend.
It's 9:30 in the morning and I am in the lab. I have planned to do an experiment this week that's called a time course. So today on Sunday I needed to come into the lab to add some drugs to my cells, and that's to prepare them for the experiment that's coming up this week. And unfortunately sometimes it's unavoidable that lab work needs to be done on the weekends, so I need to add the drug today, and then on Tuesday I will put the cells that I drugged today onto plates, and then that will prepare them for my experiment on Thursday. And then on Thursday I'll have a massive 14-hour day working with these cells, and I'll repeat that again the following week as long as it all goes well. So yeah, wish me luck. Hopefully I won't be here for too long on the weekend so I can go back home to my puppies. It's not uncommon to see other people working in the lab on the weekend. So today it appears that I am the only one in the lab. So it's 11 a.m. and I am heading home. Cells have been successfully drugged. I am keen to play some video games when I get home today. Good morning everyone. Today is Monday. It's about 10:30 in the morning. This week I have my big time course on Thursday and Friday. So most of my week is going to be spent preparing and planning for that, because it's such a big time course. I want to avoid repeating it, so I want to make sure that I do everything right the first time. So I'm also going to be preparing for maybe doing another time course next week. Wish me luck! I'm on my way to see if the cafe has sushi for lunch. I'm so excited.
5 minutes later.
They did not have sushi in the cafe. Now I am sad. Good morning everyone. Today is Tuesday. Today is the day that I need to plate up all of my cells. So today I need to plate 2 million cells per plate in 41 15 centimeter plates. I'm hoping I'll be able to take a time lapse of this, but I know the cell culture room is really busy today and I don't like to film other people who are working in the lab, so I'm not sure how that's gonna go. I'm really excited today too because one of my PhD friends is giving a really big talk alongside her lab head today. I know she's nervous, but I'm sure she's going to do a really good job, and I'm so excited for her because this is such a great opportunity. So yeah, wish me luck for today. Good morning everyone. Today is Wednesday. Yesterday on Tuesday I ended up getting my first batch of results from my colony assay, which was looking really good. My cells aren't contaminated. So I got the results for the 3,000 cells per well that I seeded. And so I realized that 3,000 cells per well is too many. So the experiment that I seeded 1,000 cells per well is still ongoing, and I'm going to check those today. But it was still nice to get some preliminary results. I think those cells had been growing for about 8 days in the 6 well plate. So today I am going to be labeling tubes in preparation for my time course tomorrow, to try and make tomorrow as easy as possible. And I also have a lab meeting this afternoon as well. I thought I would talk a little bit about what a time course actually is, because one of my friends asked me yesterday, what is this time course that you're actually doing and why are you doing it? So a time course is an experiment where you've got particular steps that need to be performed at particular times. So I think tomorrow I'm gonna have something like 12 different conditions that and so for each of those 12 conditions I need to do one thing. So I need to add drug, and then a certain time later I need to do something else. So I need to do what's called a mitotic shake-off, and then later, like a few hours later, I need to do this. And so there are set points in time where I need to perform an action for every one of my conditions that I'm investigating. And so because each of those actions takes a certain amount of time, usually about half an hour, I need to stagger each one of those conditions so that I can complete them all in a good amount of time, because I can't do them all at once. I know it's confusing. I hope it makes some kind of sense. So I will show you my plan for the time course. So hopefully that will make a little bit more sense, so you can actually see the time points and actually see the conditions that I'm doing, and maybe it'll make a little bit more sense. It's hard to explain. So why am I doing this experiment? So my lab, as I mentioned in my first video, we study defects in the cell cycle. And just as a reminder, the cell cycle is the process by which our cells divide. So in our lab, and for my PhD project specifically, I study defects in mitosis. So mitosis is the very, very specific point in the cell cycle where the cell actually separates into two separate cells, or like prepares to separate and then separates. So the time course that I'm doing I essentially have two different cell lines that I'm comparing to each other, and then I will arrest each of these cell lines in mitosis for a certain amount of time. So we use a drug that sort of prevents the cells from exiting from mitosis. So they're kind of held in mitosis when they want to divide, but they can't. And so this stresses them out, and this, they don't like it. So then after that, I will wash the drugs off of the cells to release them from mitosis all at the same time. And then we investigate how they recover after mitosis for different periods of time. So for me, I'm doing 4 hours, 14 hours, and 24 hours after release from mitosis. And then I'm going to compare those two conditions to see which cell line is recovering better and what are some of the defects that may be happening in mitosis and following release from mitosis. Yeah, so wish me luck today with labeling my tubes, and tomorrow is my big day. Good morning, everyone. It is about 8:10 in the morning on Thursday. Today is my big time course day. I have already done the first time point for my time course, which is adding drugs to one batch of my cells. So wish me luck for today. 9 o'clock p.m. and I have just finished my last time point for this enormous time course day, and I am finally headed home. I look like a bit of a mess. Check out this infinity elevator though. Good morning everyone, today is Friday. Yesterday my time course went pretty well as far as I can tell. Today I'm going to finish up my time course, so I have a few time points left, a few more samples of protein to collect. And then tomorrow, Saturday, I will need to come in to add some drugs to my cells for the time course that I will do again next week. So next week I'm doing exactly the same time course again, but just with a different cell line, because doing both cell lines at the same time is way too much for one person to do. It would be physically impossible for me to do them both on the same day. So yeah, let's see how we go. So I've just checked my cells and they all look really good, including the new 3 cell lines that I've recently made. So today I am going to grow them up into some big flasks to prepare to freeze them down next week, which will be great because then I'll have stocks of those cell lines and I won't have to make them again if anything goes wrong. Sorry about the noise, by the way. I'm sitting in front of the hood that I'm about to work in. It's a good day. So I hope you all enjoyed this week's video. As you can see, sometimes working in the lab is a really intense process. It can be really exhausting, both physically and mentally. Today's Saturday and I am so tired and so exhausted. I actually popped into the lab this morning because I needed to start setting up for my time course that I will do next week, which will be exactly the same as this week's time course, but with a different cell line. So stay tuned for next week's video for that. My time course last week went well as far as I can tell. Not next week, but the following week I will measure protein concentrations, so I'll have a better idea of how much protein I was able to extract from the cells I was working with. The colony assay that I was continuing that had 3,000 cells per well, I wrapped that up on Friday, I stained the cells and I got the results for that, and the results look really interesting, so I'm excited about that. Another thing that happened this morning was that we actually had one of our state newspapers, the Sunday Mail, they sent a photographer to our house to take photos of my partner and I and this cheeky little monkey Lucy, who, so they're writing an article on dogs who have had MRIs, and apparently they wanted pictures of this little cutie to put in their newspaper tomorrow. So if that has been published by the time I post this video, I will try and put a little picture of a newspaper clipping or a screenshot of the article or whatever in this video. So I hope you enjoyed this video. If you did, please give it a thumbs up. And if you would like to see more videos like this one, please feel free to hit the subscribe button. And as always, happy sciencing.